Hello everyone, this is your educator Priyanka Thakur and I welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. Today in this video we are going to talk about photosynthesis. Photosynthesis in today's video we will come to know about different photosystem and different experiments that were performed by different scientists. In previous video we studied about the photosynthesis pigments. Okay, what is the role of those pigments, and whatever we are going to study today you can check it out in our description box. And let's start with our photosynthesis. I haven't brought you any question today because you were not replying, but students which are watching my videos in Hindi, those were replying. Still I didn't give that question to them even, but I definitely will be asking 2 to 3 questions from them, but not from you, because you guys are not doing as much I'm expecting from you. Okay, but let's start with our lecture now. Okay, so we know that photosynthetic pigments. What is the role of photosynthetic pigment? Photosynthetic pigments absorb the solar light. We know that during photosynthesis, solar energy is converted into chemical energy. Okay, so how it can be possible? It can be possible only when solar energy is being trapped, and it is trapped by photosynthetic pigments. Okay, so now we want to talk about absorption spectrum and action spectrum. So each and every pigment will be absorbing the light from the visible spectrum at a definite wavelength. Okay, they will absorb a definite wavelength of light from the visible spectrum. Now what is visible spectrum? From 400 nanometer to 700 nanometer, it is called photosynthetically active region. Okay, the region between 400 nanometer to 700 nanometer is called photosynthetically active region or visible region. Okay, so different pigment absorb different wavelength of light from this visible spectrum, and this absorption of light of different wavelength is called absorption spectrum. Okay, so we understand now in absorption spectrum, different molecules will absorb light at different wavelength. So chlorophyll A absorb light in the blue-violet region, while chlorophyll B absorb in red-blue and more appropriately in red region. Let's check it out here. This is pigment chlorophyll A. Okay, and we can see at chlorophyll A, this is 430. Okay, this is 450, and you can see a peak before 450. This is chlorophyll A showing a peak at about 430, and again a peak on about 660 nanometer. Okay, this is 650, and this peak is after 650 nanometer. And then we can see a peak for chlorophyll B on about 460 nanometer. Okay, 463, or we can say, and for another peak that we can see for chlorophyll B is before 650, it means about 640 to 43 nanometer. And we can also see the action spectrum here, relative effectiveness in photosynthesis. So we can see that here it is showing more in blue region, violet blue region, it is showing more photosynthesis. Than again in red region we can see more photosynthesis here. Okay, so this is the absorption spectrum, this is the action spectrum. So okay, now what is this action spectrum? Action spectrum actually tells us overall rate of photosynthesis at each wavelength of light. From action spectrum we got to know that how much photosynthesis is going to be taking place on which wavelength. Okay, so this action spectrum will tell us the overall rate of photosynthesis at each wavelength of light. And action spectrum actually defines the spectrum of electromagnetic radiation most effective for photosynthesis. Okay, so action spectrum will be telling us about the electromagnetic radiations which are the most effective for photosynthesis. In other words, it is the part of the light spectrum that does the work. So action spectrum, what is action spectrum? Action spectrum, we know that we are getting the energy like our molecules, photosynthetic molecules, pigments are absorbing the energy, and by using that, by absorbing that energy, they will perform photosynthesis. And at which wavelength, how much photosynthesis they are performing, that we are going to see in the action spectrum. So action spectrum actually tell us about the rate of photosynthesis, and absorption spectrum includes the absorption of a particular light on a particular wavelength, or we can say a particular wavelength of light. Okay, and at that particular wavelength of light, there will be the process photosynthesis, and at some wavelength we will see more photosynthesis, and others we will see less photosynthesis as well. This is the Engelmann experiment. It shows us in which light there is maximum photosynthesis. Okay, in which light there is maximum photosynthesis. To prove this, he took a prism and passes a ray of light, a beam of light through this prism, and kept a spirogyra, which is a filamentous algae. It illuminates the different segment of this particular alga. And then he also used aerobic bacteria. He used this aerobic bacteria. Why did he use this aerobic bacteria? Because we know that aerobic bacteria will concentrate at the positions where there is more oxygen. So that's why, that's how we will come to know about the rate of photosynthesis, that in which light there is maximum photosynthesis. So he concluded that the photosynthetic bacteria are gathering more around blue and red regions. So it means that the oxygen is evolving more in the region of blue light and red light. So more oxygen will be evolved when there is more photosynthesis. Higher is the rate of photosynthesis, more is the evolution of oxygen molecule. That's why in blue and red region there were more concentration of aerobic bacteria, and he concluded that photosynthesis is maximum in blue and red region. And now you are going to tell me it is more in blue region or in red region. This is a question for you now. Fine. Okay. So let's talk about photosynthetic unit. What exactly is a photosynthetic unit? What we do with a photosynthetic unit? What is the role of photosynthetic unit? It is a hypothetical light harvesting unit of photosynthesis in green plants that comprises about 300 to 5000 light absorbing molecule, with a molecule of chlorophyll acting as the reaction center. Now listen to me very carefully. Actually, in a photosynthetic unit, there will be about 300 to 5000 light absorbing molecule. Now, the number of these light absorbing molecule will vary from species to species. Some species will have only 300 light absorbing molecules, while the number may vary up to 5000. Okay, so these light absorbing molecule will be absorbing the light or harvesting the light, but will have a chlorophyll. That chlorophyll will act as the reaction center. Okay, that chlorophyll is going to act as the reaction center. Now, Emerson and Arnold, they used chlorella suspension, and they also used about 24 molecules. You will read 2500 in some other books. So they used they used a chlorella suspension along with about 2400 molecules of chlorophyll, and they used the light of a saturating intensity. Okay, I hope you understand the saturating intensity. They use this light of saturating intensity for a very short duration. And by doing all this, they concluded that for about 2400 of molecule, the absorption there is absorption of 8 photons for every single molecule of oxygen. It means that for the release of one oxygen molecule, there is the need of about 8 photons of light for about 2400 chlorophyll molecules. So he concluded that for one molecule to be released, there is the requirement of about 8 photons. So according to them here, 24 chlorophyll molecules are acting as photosynthetic unit, and they will produce one molecule of oxygen per 8 photons absorbed. You understand it now? They were actually Emerson and Arnold, they themselves prepared their own photosynthetic unit in which they include about 2400 chlorophyll molecules. And for those molecules, they calculated the number of oxygen molecule released for photons. So they find out that for 8 photons, there is a release of one oxygen molecule. They were calculating all these things. So this is here the photosystems. Okay, these are all the antenna molecules which will be absorbing the photons, and they will transfer these and this energy to the reaction center. Reaction center will have a chlorophyll which further will transfer this energy, the electron, mainly electron, to some primary electron acceptor. Okay, so you understand what is a photosynthetic unit. Photosynthetic unit will have the molecules which will absorb the light. Okay, and the light they are going to absorb, the number of light, the number of photons they are going to absorb will be resulting in the release of oxygen molecule. And how, we will get to know this in our next section. So let's talk about Hill reactions now. What is Hill reaction? What is the significance of Hill's reaction? See, Hill reaction is a light-driven transfer of electrons from water to Hill reagent, which is a non-physiological oxidant. Okay, Hill reaction, in Hill reaction, there is a transfer of electrons from water to a Hill reagent, and that Hill reagent is a non-physiological oxidant. So we understand that if a Hill reaction is a light-driven reaction, then it might be possible that the light reaction in photosynthesis are going to be representing the Hill reaction. So what is the significance of Hill reaction? This reaction actually states that oxygen produced from the oxidation of water during photosynthesis occurs separately from the process that fixes carbon dioxide into sugars. So we know that there is that photosynthesis is a there occur the oxidation as well as a reductional process. Okay, so the reduction of carbon dioxide will be a separate process, and the oxidation of water molecule, which will result in the evolution of an oxygen molecule, is a different process. So this was proved by Robert Hill in 1937. What he does actually, he took an isolated chloroplast. He found out that an isolated chloroplast can release oxygen when illuminated with light in the presence of a suitable electron acceptor like ferric oxalate or ferricyanide. Okay, there is evolution of oxygen during the light reactions. Okay, so he took a chloroplast. We know that light reactions will take place in the chloroplast. So he took an isolated chloroplast and he saw that it can release oxygen if this chloroplast is being illuminated with the light, and there must be the presence of some electron acceptors like ferric oxalate and ferricyanides. Okay, so he didn't actually took the whole chloroplast, but he isolated thylakoid membrane. This thylakoid membrane lacked the enzymes. We know that the enzymes for the reduction of carbon dioxide into sugar are present in the stroma. So he took only the thylakoid membrane. This thylakoid membrane did not have any enzyme which are required for the fixation of carbon dioxide. And as a non-physiological oxidant, he used 2,6-dichlorophenol indophenol, DCPIP. It may be asked from you that what was the non-physiological oxidant that was used by Robert Hill to prove that water conversion of water, evolution of oxygen from the water molecule is a different process, and the reduction of carbon dioxide into sugar is a different process. So you should be answering 2,6-dichlorophenol indophenol. Okay, this is now we call a Hill reagent. So this reagent is blue in the oxidized form and colorless in the reduced form. Later, it was found out that the electron donor in chloroplast is NADP positive, and it acts as a reducing agent for carbon dioxide fixation in Calvin cycle. This is the Hill reaction in which water molecule will be releasing the oxygen, and 2-NADPH DP positive is reduced to 2-NADPH in the presence of sunlight. This is what you are going to see in the light reactions as well. We are not going to talk about light reaction in this lecture. We will be talking about light reactions in our very next lecture. Okay, fine. So let's talk about now concept of pigment system. How did we come to know about pigment system? We are having the two pigment system, but how we concluded that there is the requirement of two pigment system during photosynthesis. So it was confirmed by an experiment of immersion experiment on Chlorella pyrenoidosa, and he actually examined the action spectrum in the visible region for oxygen evolution. Okay, immersion, he experimented on Chlorella pyrenoidosa. Now tell me, if immersion experiment on Chlorella pyrenoidosa, then who experimented on Spirogyra, and what did we get to know about after the experiment on Spirogyra? Okay, recall all the things and answer. If you don't want to answer me, that's fine, you can answer to yourself, and that's how you will be able to memorize the things. Immersion experiment on Chlorella pyrenoidosa, he actually examined the action spectrum in the visible region of oxygen evolution. Okay, visible region for oxygen evolution. He wanted to check out the action spectrum, which will tell us the rate of photosynthesis, and the rate of photosynthesis ultimately will be telling us about the number of oxygen molecule that has been evolved during that particular process. Okay, so he found out that there is a sharp decline in quantum yield. Now, what is this quantum yield? The number of oxygen molecule produced per photon absorbed. Quantum yield is number of oxygen molecule produced per photon absorbed. And what is quantum requirement? Number of photon required to produce one molecule of oxygen. This is what we call quantum requirement, and quantum yield is number of oxygen molecules produced per photon absorbed. So he found out that there is a sharp decline in the quantum yield. Okay, this decline actually occurs at about 680 nanometer region, far red region of the spectrum, and this sudden drop in the quantum yield is called red drop phenomenon, and red drop occurs in the far red region at about 680 nanometer. Okay, this is what he found out during his experiment, that quantum yield is going to be decreased in the far red region about at 680 nanometer, and this is what we call red drop phenomenon. But in another experiment, he illuminated two beams of light on the chlorella. One is far red light, far red wavelength, more than 680 nanometer, and one is of red wavelength about 680 nanometer, less than 680 nanometer or about 680 nanometer. He found out that when two beams of light are illuminated simultaneously, the rate of photosynthesis increases 2 by 3 times. The rate of photosynthesis will increase 2 by 3 times the rate of photosynthesis with each beam separately. If you are going to illuminate each beam separately, like beam of red light and then beam of far red light, there will be lesser rate of photosynthesis, but rate of photosynthesis will increase 2 to 3 times if we illuminate the two beams simultaneously. And now this is what we call immersion enhancement effect. So red dwarf phenomenon and immersion enhancement effect were actually come into existence after the experiment of immersion on Chlorella pyrenoidosa. So this experiment suggested that photosynthesis, the appropriate photosynthesis required two photosystem, one with wavelength more than 680 nanometer and other with wavelength more or less than 680 nanometer. Clear or not? Is it clear or not? Clear? If it is clear, then it is very good for you and for me as well. Okay, I always ask you to answer your question, to post your doubts in the comment section, so that it will become easy for me to understand where are you not understanding my concept, where are you where it is getting difficult for you to understand anything. Okay, so do comment. Okay, so pigment system. Pigment system are a group of photosynthetic pigments found in the chloroplast of a plant. Okay, so what is a pigment system? The photosynthetic pigments group of photosynthetic pigments is called pigment system. In pigment system, pigment molecules are bound to proteins. Remember this thing, that photosynthetic pigments form a pigment system and pigment molecules are bound to protein. In pigment system, we will see two main parts. One is photochemical reaction center, other one is antenna complex or light harvesting complex. Okay, so one is photochemical reaction center and other one is iron sulfur reaction center. You understand my point or not? One is photochemical reaction center and other one is iron sulfur, other one is antenna complex or light harvesting complex. So pigment system will have these two things, but photosystem reaction center will again vary from for the different type of photosystems. See, photosystem 1 will have iron sulfur type of reaction center and photosystem 2 will have pheophytin quinone type reaction center. Iron sulfur reaction center is found in photosystem 1, type 1 photosystem or pigment system, and pheophytin quinone photosystem, yeah, pigment system, or we can say type reaction is found in photosystem 2. Now, antenna complex or light harvesting complex, it actually absorbs the light from the sun and transfer all its light, all its energy to the central core by the process called resonance energy transfer. These antenna molecules include carotenoids and xanthophyll, etc. But this antenna complex is absent in a photosynthetic bacteria called heliobacteria. Most of all the bacteria possess this antenna complex. All the bacteria, I mean all the bacteria which perform photosynthesis, they will have this antenna complex, but it is absent in heliobacteria. Clear? Okay, so let's talk about photosystem 1 now. Photosystem 1 is an integral membrane protein complex. Integral membrane protein complex that will be using the light energy, and it will catalyze the transfer of electron across the thylakoid membrane. Okay, you know the thylakoid membrane. So across the thylakoid membrane there will be the transfer of electrons from plastocyanin to ferredoxin. Do not forget it, that photosystem 1 is an integral membrane protein which will be catalyzing the transfer of electrons across the thylakoid membrane, but from plastocyanin to ferredoxin. So it is driven by light of wavelength 700 nanometers. So we call the chlorophyll A, which is the which represent the core reaction center of photosystem 1. We call that chlorophyll as P700, and P represent the pigment. And photosystem 1 has 700. Photosystem 1 has its pigment, reaction center pigment, which absorbs the light at 700 nanometer wavelength. So it will have the reaction center of iron sulfur So we can say it has a iron sulfur reaction center. Also contain chlorophyll A, carotenoids, and phyllo, not phyllo, it's phyloquinones. In blue-green algae, there are 11 proteins. Okay, these are A, B, C, D, E, F, not GH, then I, J, K, L, M. Remember PSA A, B, C, D, E, F, not GH, and I, J, K, L, M. There are 11 proteins in the blue-green algae. Clear? Okay. And now photosystem 2, unlike photosystem 1 which has iron sulfur system, photosystem 2 actually has pheophytin quinone type reaction center. What does it have? It have pheophytin quinone type reaction center. Okay. It effectively absorbs light of 680 nanometer wavelength, and thus the chlorophyll A in photochemical reaction center is called P680. In photosystem 1, chlorophyll A in the reaction center is called P700, while in chlorophyll in photosystem 2, chlorophyll A is called, what we call it? Tell me. We call it P680, and P represents the the pigment. P represents the pigment. In the previous lecture, I told you about the proteins. I told you that about the D1 and D2 protein and how the excited state of chlorophyll, then where there is if this excited state is not brought to the ground state, there will be the formation of singlet oxygen. That singlet oxygen is very reactive, and that singlet oxygen can damage the D1 and D2 mainly D1 protein present in the photosystem 2. If D1 protein get inhibited, then there will not be the proper functioning of photosystem 2, and hence there will not be enough photosynthesis as well. That's why carotenoids prevent the inhibition of different cellular components. So the photosystem 1 will have D1 and D2 protein. You have to remember this, and they will also have cytochrome B559. You will also know about the role of this cytochrome B559. Others, pheophytin and plastoquinone and other cofactors are also present in the photosystem 2. Distribution of photosystem 1 and 2. Photosystem 1 is actually found in all the non-appressed members of the grain and thylakoid. Let's check it out. See, this is the non-appressed part. Black one is the non-appressed part. It is not appressed. So in all the non-appressed part, even in the stromal thylakoid, or we call it a stromal lamellae or thylakoid lamellae, photosystem 1 will be present, while in photosystem 2 is found in the appressed part of the thylakoid membrane. So you shouldn't forget about the reaction center. Iron sulfur type of reaction center is found in photosystem 1, while pheophytin quinone photosystem is found in reaction center is found in photosystem 2. The chlorophyll A is P700 in case of chlorophyll photosystem 1, and chlorophyll A is P680 in case of photosystem 2. The D1 proteins are mainly present in the photosystem 2, and cytochrome B559 is also present in the photosystem 2. And photosystem 1 is found in the non-appressed region of the thylakoids, while photosystem 2 is present in the appressed region of the thylakoids. That's all for today. We will meet later in our next video. Do comment down in the comment section to tell me about your doubts, and subscribe to our channel. Press the bell icon if you want to get the notification of each and every of our video. That's all. Take care. Stay safe.